Hello, Dr. Jacob Worth.
Hi.
You are Dr. Jacob Worth.
Yes.
Again, this week.
I would say still.
Really?
Yeah.
You don't feel like you ever change it up and come back to yourself?
Well, I definitely change it up, but I think legally I'm still Dr. Jacob Worth. What's going on, buddy?
Can you tell me about this week's group meeting? About this paper that came out? What's its title? What's it about? Who put it out?
I caught most of that. I was distracted. But yeah, I can tell you about the paper.
So that I can get lay.
Get lay.
Let's get lay.
That's cute. So this paper, the title is The Molecular Basis for Branched Steviol Glycoside Biosynthesis, which, from the get-go, is just what is actually needed on the molecular level to make these sugars, these steviol sugars, the glycosides in the body. And steviol glycosides are familiar to most people because brands like Stevia that make non-caloric sweeteners.
I know stevia.
Yeah.
I've had a stevia inside me.
A stevia once or twice.
Very bitter.
So that's one of the things is there are a lot of steviol glycosides because there are a lot of arrangements you can put glucose sugar molecules onto the basis, the scaffold molecule. And some of them are bitter and some of them aren't bitter. And all of them are kind of sweet, but they're different levels of sweet. Now, one of the ones that we use regularly, that's pretty much like the one, seemingly is called Biosynthesis A. So there are a couple different steviosides, and one of them is like the go-to. This is what we make as a non-caloric sweetener.
If you commit stevioside, do you go to jail?
No, only if it's some of the steviosides. There are many types. And I guess just a non-caloric sweetener, for the record, is something that can make the taste receptors you have tell you it's sweet, but you don't actually break it down in such a way that if bonds aren't being broken and reformed as it goes through your digestive system, then there's no energy really being released. So you're not deriving any sort of calories from it because you're not actually doing anything chemical. So something like Biosynthesis A, when you put stevia in your coffee or your tea or whatever you use it for, it just goes right through. You don't actually use it metabolically. It just binds to some receptors at some point, tells them they're sweet, then it's released and it leaves. So that's how non-caloric sweeteners work. And there are a couple different ones. One of the things about the stevia-based ones is that a couple of the steviosides actually do have this weird kind of bitter stevia-esque, like very specific, you can tell it's that sweetener aftertaste.
Yeah.
So you have things like the mogrosides from monk fruit.
Ooh, I use a lot of monk fruit.
And that has a different flavor, just like sucralose and Splenda has a different flavor, just like Sweet and Low or something. Like all of these. Mm-hmm. Is that a monk fruit?
That's sucking up Lakanto.
Oh, okay. A single monk fruit. All of these have different tastes because they're all different non-caloric sweeteners. So stevia ones you get them from plants. There's a stevia plant. I forget. The Stevia rebaudiana. Okay, that makes sense. That they would be steviols and rebaudiosides. Stevia rebaudiana is a plant, and that's where we get the molecules from. Because we don't really make them. We can't make the molecules very well on our own. Especially because the biosynthetic pathways that make some of these molecules, we don't know all the steps. We know that, like, if you start with this, the plant will make, if you give it A, the plant will make B, C, D, E. And we know E, and we know how D, and we know how D goes to E, and we know how A goes to B. We have no idea how anything makes C, at all. We kind of think we know what C is, but we don't know what makes it, and we don't know how that goes to D. We have no idea. So you can't remake these pathways in the lab. You can't do this industrially because we don't know how nature does it. So we just culture the plants and then extract it from the plants.
All right.
Which, isn't the most efficient, but...
Do you know how we discovered this plant?
This plant?
Yeah.
I don't off the top of my head. I would promise you it's native to Paraguay and Brazil. I promise you this has been used by indigenous populations of South America for hundreds of years before European colonialism ever found them. I guarantee it was just a thing used to make stuff sweet. Because that's how a lot of these things, I don't know if that's actually the way, I don't know the story, but a lot of times
But I do guarantee it.
I
don't know, and there's no way for me to find out right now.
But
I will give you that guarantee.
But I promise you, it's something like that. So one of the problems with getting it from extracts is if I get this plant and I powder it or whatever, or blend it, or however you want to do the extract, and I add solvents to it and I extract some of the things out. Let's say I can get rid of all of the solvents that I use, so it never goes into a person because the solvents are bad, which is already a promise that's hard to make.
Yeah. Okay.
It's already not the best promise because that's actually one of the things that you run into with marijuana strains is the solvents are caustic, and because it's been an illicit substance for a while, people don't treat it properly, and so you can get sick because the extraction process was bad. So we have these crazy high-tech extraction processes because this has to go into humans, but the predominant steviol glucosides are stevioside, which is 5 to 10% of the dry leaf weight, which means 90% of the leaf that you are harvesting is useless to you. You have to get rid of it, which A, is wasteful, and B, you can't promise that you'll get all 10% of that when you extract it. A bunch of that will go to waste just because your extraction process isn't perfect. So you're dealing with so little.
Have we tried feeding it to the pigs? The
plants?
95%.
To see if they make Rubatioside A? Or just because?
No, just because you don't want to waste it. You have all this product.
I'm sure they use it for something. I would guess, use it for fertilizer, right? And it's plant.
Feed it to the pigs?
Plant matter. Yeah. Pigalizer.
It's pigalizer.
Here's the number I wanted. Rubatioside A is 2 to 4% of leaf dry weight. But they taste 300-fold sweeter than sucrose. You can add less stevia to your coffee to get the same sweetness as if you were adding sucrose.
Have you ever seen what it takes to make sugar from sugarcane?
No.
It's an insane process, and it takes very little of the plant itself. Most of the plant is a fibrous husk.
Yeah. Yeah. That's the problem. That's so wasteful to need to do that. But humans want sweet things, so that's what we do. But wouldn't it be cool if we didn't? So...
can we do that? Can you just don't want sweet anymore?
Oh, no. I'm saying, wouldn't it be cool if we didn't get sweetness that way? I think we're going to need sweetness, so I would like to do it in an environmentally sustainable way.
No, let's just make humans not like sweet things.
Well, that's not what these people did.
Oh, okay.
So, one of the problems that they set out to look for is, A, for the Rubatioside A, the sweet thing, that actually is the less bitter after-tasting one. Because stevioside is, that's the one that even sounds like stevia, right? But that's not, the primary non-caloric sweetener that tastes the best derived from stevia. That would be Rubatioside A, but nobody knows that name because it sucks. It's just a weird chemically name. But stevioside, that sounds cool. But basically, Rubatioside A is the one. And we don't know all of the things that make it. But one of the really cool things is Rubatioside A has a branch. So it has the main core hydrophobic natural product structure that it's got a lot of rings and carbons and it doesn't like water and it's not very soluble. And it's a terpene. Not that that matters, but that's one of the biggest class of biomolecules. So it has that core. And then it's decorated with different sugars. And that's how these things kind of work. And that's how the sweeteners work. And that's how a lot of natural products work, is that a lot of them look the same. And then you have a bunch of extra bits tacked on. So with this, it's an extra couple glucose molecules that are tacked on. But with Rebaudioside A, it doesn't just make a chain, it makes a branch. And it has to make that branch. And it makes that branch specifically very efficiently. That you don't have different, like the pathway is designed to make this branch. You don't ever not get the branched sugar by the end. So that's difficult for a couple reasons, even though that sounds kind of on the face like it might be easy or simple. But when you think about it, A, you have to put the first thing there. If you're making a branch and you're making a Y, you need the stem part of the Y. That has to go there first. So something has to do that. Then something has to add either the left or right branch. And then something has to add the other branch. So not everything can do all of those jobs. It's going to require different enzymes because one that adds the first one, the stem glucose, has to find a big-ass natural product scaffold. Whereas the other ones have to find a glucose because they're tacking glucoses onto each other. So they have to find very different things of this molecule to do this pathway. The steps are not, so it's like add a sugar. Sounds easy, but they're adding a sugar to a very, very, very different thing. And therefore, you need an entirely separate enzyme to do it. Which is why you have these pathways that have like 50 steps. What they didn't know is no one knew before this paper what was responsible for adding the chain. Because the chain is what makes it Rebaudioside A, and the chain makes it really sweet and not have a bitter aftertaste. And the chain that branches in this way is what makes it a good non-caloric sweetener. But nobody knows why it branches. Except for them. And us, now, because they made the paper.
Well, I don't know.
Mm-hmm. So, all of that is just the foundation of why anyone cares. From that, do you have any questions?
Not really. I mean, it seems kind of complicated in general, but that seems to be the point of this study. It's a complicated way to make it sweet, and they found the best path to it.
Well, they have a question mark on one of the arrows. In figure 1A, and they're drawing the pathway. You can see here, they go molecule, arrow, molecule, arrow. Above every arrow, they write the enzyme that does that step, except for right here, and they go UGT question mark. Whoops. I guess we don't know that one.
Did they talk about it?
No, it's just not known.
Okay.
They did not set out to elucidate this pathway because they are not cell biologists who would probably be cell and molecular biologists would find that pathway. They're biochemists and do a lot of what I would do, which is they take a step of that pathway that looks like it's magic and they figure out how does that happen though? Because if you have your big ass molecule, what?
I do have a big ass molecule.
And then you add a sugar to it. And then you get another enzyme to add a sugar to that sugar. That enzyme, that other enzyme, the second one, conceivably could just add one and then add another one and add another one and add another one. This step
But he's tired.
Well,
he already did his job.
You would get other buddies, his twins.
Oh.
His siblings would keep adding one more because you would find sugar, add a sugar, sugar, add a sugar. Here, you have a very specific enzyme that comes over and says, oh, there are two here. I need to add one, but I need to add one in between them. And I don't need to add to the end. So that is where this, and people like me come in and go, okay, but how does it add it to the middle and not to the end? Because it's adding to a sugar. It's adding to the stem one. But it's not adding to the arm one, ever. You don't find Rebaudioside A, but with 3 in a row. You find it with the Y.
Stem meaning towards the molecule itself?
Yeah,
meaning
the big scaffold.
Towards nothing?
The big scaffold. The stem, so you'd have the big natural product scaffold, then you'd have a stem, and then you'd have the branches of the Y.
Okay.
So it adds to the stem, which is a sugar. It's a glucose added to a glucose. But it's only that specific glucose. And it has to already have the second one there. Yeah, I know. So that's right. It's kind of weird. So that's what they set out to learn. They didn't seek to learn what this step was. That's somebody else's job. It's also an interesting question because we know this, and especially because anyone who's taken an organic chemistry class, like there are questions on tests where they basically give you a molecule and then they give you a second molecule and they say, what is the name of the chemistry that happens here? And you have to look at like where the bonds go. Yeah, it's Timothy. Timothy chemistry, his name is. So we actually know the chemistry and the rearrangement of the molecule. We know everything about what happens here. We've just never found the thing that does it.
Okay.
But we did find the thing that does
do we need to find
the branch. If you wanted to do this from scratch, yeah. But in an industrial setting, like where what happens at my work and companies like ours, is you basically have to say, like, let's say I don't want to culture this from plants because plants are hard. And I want like yeast to do it because yeast is easy to grow. You can only put so many enzymes, new enzymes in the yeast because the yeast doesn't make steviosides on their own. Otherwise, that would just be stevia. Yeast don't already make it. That's only this plant makes it. So you have to figure out, I can only add so many new enzymes to yeast before it stops wanting to work because it has too many other moving parts and you're screwing with all the other pathways in it because yeast doesn't want to dedicate energy to making stevia. So you can't add everything. So you have to kind of figure out, okay, well, what's the cheapest, easiest precursor I can start with? And I can feed that to the yeast and they spit out the end. So on the quest from A, B, C, D, and E, maybe I don't know how to get to C. Maybe I can't add stuff to C. But I can make C, D, and E in a yeast, and that's easy. So I'll just buy a bunch of C, and I'll just only feed it C. And I'll let nature give me A and B, and then that's fine. And then that's kind of the trade-off of you can't start from scratch for engineering a pathway, especially if you don't know all the steps. But you can start in places. Right now, the plant does all the steps. If we can design some of this pathway to be not just the plant, at least for some of the steps, it becomes easier to do the steps. But we don't know how to make the branch. So they are a pretty straightforward biochemistry and X-ray crystallography lab. So a lot of their figures are kind of boring if you don't know what you're looking at already.
Big ribbons.
They have, yeah, it's big ribbons. They have a ton of protein. I mean, there's bar charts and stuff later.
Listen, I've seen a lot of big ribbons figures in the, what was it, 4 years or 5 years that I witnessed your going through grad school. I've seen you make a lot of ribbons.
You saw 5, yeah.
I don't know what the hell I'm looking at.
Yeah, and that's fine. So basically, the way the figures work is an amino acid is a molecule, right? It's just a molecule like anything else. So
I'm with you.
If you have ever seen someone draw a molecule, it's like little sticks, and sometimes there's letters like N or C or O or whatever. Amino acids are just those. But when you put a bunch of amino acids in a row to make a protein, they start to adopt very specific patterns. And they do that because all the amino acids have something different about them, which would be their side chain, but all of them have the same main chain, and it's the same set of atoms over and over and over and over again, and that makes the backbone of a protein. So the backbone can only adopt specific shapes because of the laws of thermodynamics. Like, it's just, they have to make these shapes. So you only ever find a handful of repeating patterns in a protein. So because of that, if you have a bunch of amino acids in a row and you're looking at the protein in some molecular viewing program, if you have a bunch of amino acids in a row, you can basically just collapse them into a specific representative shape because you've looked at protein structures before, that you don't need to know where every single atom is because it just makes that shape. And it's basically helices and sheets, that they make flat sheets or they make spinny helices. So basically, you can just look at all these structures and say, oh, look at all these helices and sheets on top of each other. And then when I say, oh, well, this alpha helix does this thing, you don't have to search for a handful of like 100 atoms. You're just like, oh, that little tube? Okay, that tube does this thing. That's a tube. And then when people like them and like with figures I've made, you choose one or two of the things on that tube and you put in the actual lines and atoms of that thing. Because everything next to it doesn't matter. But the actual atoms of that amino acid are doing something special. So figures like this, like figure 1b, basically it's just a bunch of tubes and sheets.
That's not a single bee. That's a bunch of ribbons.
There's also sheets.
Are there bees?
There's a couple bees.
Bees?
Look. Well, that's a beta. Beta counts as bee.
That's not a little honey bee.
That's a bee.
There are bees.
There are bees.
But it's not one bee.
There's not only one bee, but there are very little. Or few. The point is, they have ribbons and they have sheets, and then they have the actual molecule that it binds. And the kind of protein that adds these sugars are called UGTs. And the only thing that is important about them is that they take a nucleotide called UDP that has a glucose on it. It's called UDP glucose. And they add that glucose to some other substrate. And that's their whole job.
What's a substrate?
Substrate is the thing that the enzymes like to use. There's the thing they use.
The tools. Enzyme tools.
No, because enzyme tools would be like the piece of the enzyme. It's like if the enzyme is designed to make a product, the substrate is the ammunition. It's what it turns into the product.
So the materials.
Yeah. But, so if you're saying this enzyme makes ribadioside A, that's the product. But there's the thing that brought the sugar and the thing that used to be something that turned into ribadioside A. So the product's ribadioside A. But the thing that brought the sugar is also a substrate. And the product besides ribadioside A is UDP because it's the shuttle that brought the sugar over. You also have to spit it out. Like, that's part of the reaction because that's how reactions work. So both are substrates. But when people are talking about like the enzyme product, they're usually or oftentimes talk about like the point of the enzyme. The enzyme's point isn't to make UDP. It makes it, but only kind of because it uses UDP as ammunition for something else. So I guess it's jargon and nomenclature, and it comes down to the substrate is, it's the building materials. But you don't build on top of it. It's like you transform it into something else.
It's the lump of clay that you turn into the vase.
Yeah. Yeah, sure. We have to be careful with that because the rest of this analogy has to carry the rest of the paper because a lot of it is talking about the active site and how it holds the substrate.
Oh no.
So if you're ready for lump of clay...
Does lump of clay work?
It might. I guess we'll see.
You're my beautiful lump of clay. My substrate baby.
Substrate baby. Substraby?
Substraby.
That's below the normal straby.
Yes. You are worse than the average straby, but I love you anyway.
Substraby. So they used X-ray crystallography to find the structure of the protein. Nobody knew the structure of the protein before. They figured it out the first one. Awesome. And they got it bound with the substrates in it. And that's not trivial because you can get the structure of a protein, but if it's not bound to the thing it uses as ammunition, maybe that's not the shape of the protein that you care about. Maybe that's not... normally it squeezes or twists or turns or something when it binds the substrate. So if you can't get it with the actual substrates bound, you basically can't learn a whole lot about it. So they were actually able to do that, and that's really cool. And they go on a little bit about how that's neat, and because it's an X-ray crystallography study, you can't ever promise that you can do that because sometimes you just can't. And they identified a bunch of amino acids that rest up against the sugar molecule and the, what do they call it? The steviol, which is the scaffold molecule, the natural product that we're starting with. And you can see in figure 2, they show the density map in 2A, which is basically, this is from their crystallographic studies. This is the shape that their program spit out and said was inside the protein.
That's a nice shape.
And you have to put in your guess at that molecule of what that shape is. You're given basically it's a wireframe. And then you have to put in all the molecules. And some of that's easy because it's amino acids and they always have the same atoms. And they always do the same patterns. But basically you get a blob wireframe and it says, well, this is in the middle of the protein and we know it's not connected to any of the other parts of the protein. So it's its own separate entity. And we go, okay, well, geez, I really hope it's UDP. And then you try to model UDP in there and it fits exactly. And you're like, okay, cool. So
magic.
So they're showing, the only reason they would show this is they're saying when we say this substrate's touching this amino acid, we are not basing that on nothing. Look at how good our wireframe is. Look at how good it fits in that wireframe. All of our conclusions that follow about what the amino acids do are based on the fact that that shape is complete. Because sometimes you have them like peter out and they have like little non-shaped blob droplets over here and you're like, oh wow, that part didn't resolve, but this other part did. So I can't promise you in that little blob droplets any interactions. If I try to claim that any of those interactions happened, I'm probably lying. Or if I'm not lying, it's because I got lucky. Like you can't know because you don't see where the molecule is. So they're just showing the very impressive, if you've seen electron densities in crystal structures before, they actually show a very impressive electron density that they really do know where the substrate is. It's actually a really well-resolved crystal structure. And the resolution is okay. Oh, actually, they got two structures, one with the UDP and one with the UDP and ribaudioside. So they actually can see, this is what happens when the sugar, the transportation thing for the sugar, the part that's bound to the sugar molecule, UDP, they have it with just that, and then that plus the ribaudioside A. So they can actually see how the enzyme changes when it gets where the sugar needs to go and where the sugar can start. So, you know, the enzyme has to change at some point between those two steps because it has to take a sugar and put it somewhere else, which means at some point the enzyme does something. So if you can get structures with one substrate bound and then both substrate bound, you can get snapshots of how the protein changes. Which helps because if you're trying to construct an entire story from one snapshot or Polaroid, it's hard. But if you have a series of them, you can now say, oh, well, this is kind of the way things had to move, and this is actually the narrative of whatever happened here. Whereas if you do everything only on one snapshot, you have to make some assumptions. So that's neat. I didn't see that they got 3 structures altogether. I thought they just got one and then basically a confirmation one. I didn't see that they got a 3rd one. Oh, neat. Not that it really matters for most of their figures because it tells them more or less the same information. But what they find, and in figure 3A, they show the whole main idea of why this is neat. And they show 2 UGTs. They show 2 enzymes that add a sugar and then a sugar to the end of that sugar. So those are the first 2 steps, that you have the main molecule, you have steviol, and you add a glucose, which gives you steviol monoside. Then you add a sugar to that sugar, and that gives you stevio bioside. Then you have a separate one, add a sugar another place, not to that chain.
Stevio in betweener side?
No, not yet. That's...
on our underside.
No, that's just called stevioside. So we have stevio monoside, stevio bioside, and then stevioside. So it's like they gave up adding little qualifiers.
I think they should have done on our underside.
Yeah, stevio on our underside. And actually, if you look, like, I don't know if you can really see it, but they're drawing this chain here.
Yeah.
And then this one is just right down here. So it is on the underside of the molecule for no reason.
It's on the underside.
But it's neat because things in that pathway, you never find this on the bottom there until you have this chain. Mm. So like, it never shows up when there's still only one molecule. You need 2 first. And then you never see the branch happen until this one's on the bottom side. Even though the bottom side isn't part of this main chain, this never branches unless that's there first.
The old never branched.
The old never branched. So that's the kind of reason, that's the kind of thing that gets people like us excited is,
no, is it?
Why does a different part of the molecule need to have a glucose on it? That's not involved in the chemistry at all. It's required for a glucose to happen over here. It doesn't make any sense. The problem is that it does make sense. We just don't know why. So that's the kind of thing that makes people like me excited. Is
No, does it?
why does a molecule need to be way over here for chemistry way over here to happen? Like, they're not related at all. But there's some biological reason that happens, and that's why it's kind of neat to find out. So what they find, the ultimate question isn't just how does it add a sugar to in between to make it a branch. So you're adding a left branch instead of a third in the series. Like, that's the big question, is instead of making 3 in a row, we add 1 to the middle of those 2 to make a Y. And that has to happen for some reason. And they find the other UGTs in the pathway have a lot of the catalytically relevant amino acids, the ones that are actually responsible for doing the chemistry, the actual sugar transfer. All of them are conserved, which makes sense because if the general family of enzymes is doing the same chemistry, having them all have the same chemical step of these residues, these amino acids, shift the sugar this way. Those all being the same, the ones that are actually like pulling and pushing, that makes sense. All of the other amino acids that aren't the same are probably dedicated to just making them selective, of choosing the right substrates. Because if you had every UGT able to add sugar to every single thing, your body would just be filled with with sugars on them, and you'd never have anything else. So everything has to be really, really selective.
I would like to put forth that my body is filled with things with sugars on them.
Yes, but not exclusively.
You don't know what I eat.
That's true. I mean, I know a lot of the things you eat.
We do live together.
Yes. But I guess I'm not always here. So what they find is there's the main pocket, and they show the cartoon of this in figure 3A. And they show the actual ball and stick figure in 3C and D, and 3A is very much easier to follow. What they show is that in this special enzyme, UGT76G1, which is not an important name, it's different than UGT85C2 and 74G1, which is also not important names. But we have the special one and the not special ones, are basically what I'm going to call them. Now, we have to go to your clay pot hand analogy, okay?
Okay.
So you have...
Do you want me to be Patrick Swayze?
Not tonight. I have a headache. So...
You always have a headache when I want to be Patrick Swayze.
Maybe there's a hint there.
Do you want me to be Whoopi Goldberg?
Never. Not any night. Not any headache. So when you have your clay, do you mean like on the little spinny thing where you have to like shape it with the clay that spins?
Well, it needs to be for the Swayze thing, but you're not going to do that, so you can do it any direction you want.
I mean, no, that's fine. So you want to make a vase, right? You said a vase.
I said a vase.
So you hold your hands in a special way as it's spinning, and you're basically making the central core that is going to be a big hollow part, because vases have hollow parts, right?
Yeah.
And generally, not all of them, but a lot of them.
That's why I feel akin to them.
Yeah. Wait, because the flowers?
Because I'm empty inside.
Oh, okay. Not all vases have like a big chamber and then like a neck. Some of them are just like all one tube. But we're gonna pretend we're talking about a vase that's like a big chamber and then it has a neck.
I like to think of myself as all one tube, so I don't get to think of myself as this vase now.
That is fine and was not necessary for the analogy. I like that you brought it in, but I'm not that upset that you have to remove it as part of the equation. So you hold your hands a certain way to make the big chamber. And then you hold your hands a certain way to make the neck, and you shape the neck a certain way. Mm-hmm. So you have enzyme A makes a big chamber and then makes a neck. Then you have enzyme B has a big chamber and a neck, but it wants to make that neck longer. So let's say instead of starting from scratch, you just take the thing from A, and you're like, okay, cool, that's fine. I can just hold my hands in the same way. I make the same shape, and then I just make it longer at the end. The important part there is that your hands for the big chamber are the same because you're setting up the same shape to go into a neck. That's the same neck, just longer. Enzyme 3, which is the special one here, can't hold the main chamber the same way because if it does, you have set it up to make a neck of a very particular shape. You don't want that shape. You want a different shape. You want the magic branch. So,
I want the magic branch.
Even though it's grabbing the same chamber that the other UGG's grabbed, which is that natural product we keep talking about, the scaffold, the big part of the molecule, that's the real molecule, that isn't just a sugar chain.
Big Daddy.
Big Daddy. It has to grab Big Daddy because it has to bring it in to bind it to do chemistry because it's an enzyme. Mm-hmm. But it has to grab it in a different way so you don't set it up to do the same thing. So they see, and they kind of do, it's really hard to see if you're not used to protein structures in the PyMol program. It's hard to see with figure 3, C, and D. But in A, they do remember last time where I said the cartoon that they tried to summarize everything in was the worst cartoon ever?
I remember that. You were very mad.
This cartoon is great. This is exactly how you want to make a cartoon. Because you could have made this in PowerPoint in like 5 minutes.
If it's exactly how you should make a cartoon, is it the Venture Bros or is it She-Ra? The two perfect cartoons.
It's probably She-Ra because it's more approachable as a cartoon. Well, it's hard to say. It's hard to say.
Which I am.
Yeah, but She-Ra is acceptable for all ages, and Venture Bros is probably lost on a 5-year-old. So actually, it's probably closer to Venture Bros, because you do have to know a little bit of what they're showing you. Basically, what they are showing is there are 2 alpha helices, 2 of those big tubes from the structure, and normally they kind of rest parallel with the thing squeezed between my fingers and my palm. With the special third enzyme, it's twisted, so instead of being this way, they kind of twist so they align, the palms align. Mm. And now, this...
Holy Palmer's Kiss.
So instead of doing this with your clay pot, you're just doing this. And that's going to change everything that follows, because you've made a completely different setup of shape.
That's like ballroom dancers. The way they'll touch each other to initiate a move is very similar, but they only need a tiny little difference of touch in order to know what move to go into.
I didn't know that, but yes, it's exactly like that. It is precisely like that, which is really cool. Because sometimes it really only takes a little shift. Because as soon as it shifts like that, the little oxygen over here, the part of the stem sugar that has to get a second sugar added to it, is now, instead of positioned like forward, it's positioned slightly out. Which makes it exposed, which makes it able to have a sugar added to it, and otherwise would not have been available to add another sugar to. That's just one part of the equation. So you have to set it up, which it did. But then they found another part that, not special protein A and B, have a series of amino acids where that, so you've kind of cocked it one way, like an angle. But you have a sugar here, right? Because this is a branch, so you has to already be 2. Mm-hmm. So you've cocked it a little bit of a way, which means that second sugar that normally is being added by something else, if it's already there and cocked to one side, you need a place to put it. And the other enzymes just don't have it there.
Well, if it's already there and cocked to one side, I know of a place to put it.
Well, it's got to be squeezed in there, but it has to be done in an energetically favorable way. Otherwise, it won't stay. And if it doesn't stay long enough, you can't do the chemistry.
Oh. Yeah, the place I was thinking was not energetically favorable.
No, that's the magic of enzyme kinetics is...
Lots of expenditure.
Yeah. And generally, if there's a payoff at the end...
Well, there's a payoff.
But a payoff for all parties.
Just for me.
Well, then it's impossible. They have this The special one, 76, has a handful of mutations there that pretty much universally are all the same kind of amino acid, but smaller. So they're just less bulky residues. So instead of having an amino acid with 4 or 5 carbons as a side chain, it's got basically the same shape, but 3. Like really subtle differences, but it has a handful of them. So you slowly see a tiny bit of a pocket that's normally not there, just widened enough. But the overall chemistry of that pocket, everything going on inside of it, is the same. But now it can fit a sugar in it. So it cocks to one side, shoves this sugar somewhere that only this enzyme has, and now you have an oxygen just kind of floating around. And a substrate kind of locked into place. Like the enzyme is now holding it in 2 different places. It's got a favorable pocket for the sugar, a favorable pocket for the scaffold, and an oxygen pointed right into the solution, or right into the open solution part of the active site. And that's where you can have UDP glucose come over and add to it.
What's an active site?
The active site, that's where the enzyme binds something to do stuff. It is exactly what it sounds like. It's not a fancy jargon at all. It is the site of the protein where activity happens.
Yeah, where there's enough space to do activities.
Yeah, basically.
We've got the bunk beds.
Mm-hmm.
We have an active site.
And you have to be careful with an active site because you have to have it just closed off enough that you can hold on to stuff, but just open enough that things can get in there in the first place. So like a kid's, like a playpen. If it's too small, you've done a good job holding the kids, but they can't move around. But if you make it too big, you essentially don't have a playpen. They can do whatever they want because you've made it the whole room. So you have to be really careful. And that's essentially, this enzyme, it opened up a tiny bit of the playpen in one corner, so now kids can go there if they want, and no other playpen on Earth has that. And then the sugar can get added. So all of that, if you don't look at the structures or the cartoon or understand really what they're saying, all of that is an incredibly boring way to say it holds it in a different direction and then grabs onto its arm to not let it go. And then it can add a sugar to a place that's normally not available to add to. Normally it's covered up. But with 2 subtle changes, this protein now completely exposes an oxygen that normally is not available at all. And it can make a branch. And that branch, for whatever reason, touches the receptors in your tongue and tastes much sweeter with way less bitter aftertaste than other parts of the stevia plant. The branch is magic. I don't know what the branch does. I'm sure there's another paper there where people describe how the branch binds to those receptors. Maybe there's not. Maybe we don't know. There's lots of stuff we don't know. But we know that that branch is important. And then the final part of their paper is essentially figure 4 and table 2 is they find all of the amino acids that they think might be important and they mutate them to other things one by one and see if that breaks the enzyme. And I can tell from this figure and this table, this is a person's thesis. This bar chart is someone's 6 years of work. Because they got that, which is...
So it took them 6 years to make one bar chart?
Just about.
They must be slow at making figures.
That's... they're not very proud of that student. Unlike the last paper that had a bunch of different kinds of experiments and 30 people, this has 4 people, and I guarantee Soon-Gu Lee did almost all of it. The first author. Jez didn't do much because that's PI, so he did like overall guidance and helped write it. Yeah, he's a Jezebel. I would guess, just eyeballing it, that Solomon was a younger grad student who helped, and Oliver Yu was like a tech or like an undergrad person.
Oliver Yu?
Oliver Yu.
He did that on purpose.
He made his own name on purpose?
I mean, maybe. If Yu is a Chinese name, then a lot of Chinese students, when they come over, have a more...
Yeah, it's true.
He could have...
Yeah, he could have made it Oliver Yu.
What I'm saying is Oliver Yu is my hero.
He is likely an undergrad that helped and got into a good graduate school because this is a solid paper that does really good work, and if he was a part of it. Now, all of this, it could have been 3 postdocs, and maybe I'm wrong, but this paper feels like a paper that could have come out of my lab or my department, this feels like it could have been someone I know. It's a very straightforward, to me, study.
And Oliver could have been his real name, and his parents just have a great sense of humor.
I hope so. I hope it's that one. And he just rolls his eyes. So they go through and they mutate all of these things, and they mutate them to chemically similar ones, and then also mutate them to an alanine. And if you mutate something to alanine, you basically, that side chain is just a single carbon. So what you're saying is you're going to mutate it to something that doesn't affect the overall structure of the protein, but you're going to take away its ability to do chemistry. So that's like the magic test of, because I could mutate something into something huge and make the protein fall apart. And it doesn't mean I know anything about the chemistry. It just means I made something fall apart. So if you mutate to an alanine, you've taken away its chemistry, but you haven't messed with the backbone of the protein. So ideally, you're not doing anything like that harmful, unless you're taking away part of the chemistry, and then you can kill the enzyme. So that's the test. So they mutate a couple things to alanine, and then a couple things to chemically similar things, like change a positive charged amino acid to another positively charged amino acid. And that way you're saying, okay, their shapes are different, but the charge is still there. And that test is basically, is this charge important for the chemistry, or is the shape important for the chemistry? So they do a couple, handful of mutations for each of these spots and come up with a general set of what they think the important ones are. Some of them kill the enzyme entirely. Some of them just reduce the activity a lot. But the positioning of the core, you can't change. That's important. The cocking it to one side, super important.
Gotta cock it. Cock and lock it.
But all the things binding and associating with the glucose that they had to make a new pocket for, all of those are essential. If you get rid of them, it breaks.
I think the binding I already put forth in the locket.
Oh, yeah, that's true.
Yeah.
You know what?
Being redundant.
That's true. If you take away some of the chemical interactions or make things too big in that new pocket, you break it, which is neat because that means it's really carefully designed by nature, which is neat. Whereas the residues that make up the branch that set up the new glucose, you can actually mutate a bunch of those and you make the enzyme less active, but it's actually perfectly viable enzyme even with that. So where you put the new branch is not nearly as important as what you do with the old branch while you do the chemistry. And that is part of the mechanism that's really important. That's like a big take-home message is that means, ideally, if it doesn't really matter where we're putting the new thing or how do we hold it or how we put it on, as long as we design an enzyme that holds the first branch there, we can add whatever we want to the second branch. It doesn't need to be a glucose. It could be anything. Because we know the enzyme doesn't really care what goes there. Which means if I want to create glucose and galactose, or glucose and mannose, or glucose and any of the other things that could be sweeter, or easier to make, or cheaper, or quicker, or anything, we now know something about the mechanism that we didn't know before. Because at first glance, if I asked you, what are the important parts of the molecule to hold? There's no reason to not assume they're all basically equal. But now there is reason to assume, because some of these bars are still bars, and some of them aren't there at all. They're just baseline. So, that's a thing. And then they have the individual parameters on when a mutation happens. Does it affect the speed of the reaction or the binding affinity? And some mutations affect both, and some only affect one. And there's probably projects in this lab that are going on right now figuring out well, why does this one affect the speed, but this one affects speed and binding capacity? That doesn't make sense. Why would you only affect one? So there are reasons, I'm sure, that they have as guesses as to why. More or less the scope of this project was to figure out how on earth do you make the branch in the first place? Why do we not know how this happens? And now we have a pretty good at how it happens. And the big take-home message, the big one, is they also know what mutations were necessary to make the new pocket, right? They already figured out which ones were necessary. They already figured out that what you can change for chemistry and what you can change for size. They already figured out exactly kind of the what you can do and still adequately make the branch happen, right? Don't look at me. I can't do anything. You're over there. So if that's true, you should be able to take a different UGT that doesn't make branches and put in the mutation. And the obvious next study for this that I imagine, knowing from what I know about PIs, I'm almost positive the PI wanted him to do that before he left. We're like, oh yeah, but before you graduate, can you... So if that's true, and I'm sure it was part of his project that he didn't get to or he didn't want to do before he graduated or something, take one of those other enzymes. So when you go to the mutational studies, first thing you do is, I think these amino acids are important. Let me take them away and see if the enzyme still works. Oh, when I took them away, the enzyme no longer works. Clearly they're important. The second step is now what? Can you guess?
Party.
Yeah, you party, pop champagne.
Yeah.
You graduate, you move on. What is the next part of the study? I take the amino acids away and I see that they were important because the enzyme breaks. In my quest to say, how are branches formed? I now just told you what amino acids are needed by the enzyme. What would you guess the next step is, besides party?
For what end? What are you trying to do?
The ultimate question, the thesis question is, what does it take to make a branch? What does it take to make a branched glucoside?
Can you have there be less of the enzyme to see if it still does it?
There's that, and that would be more of a pathway question. That's a good pathway engineer question. I mean, I assume I'm not a pathway engineer. That's more along that line of like what's necessary from the pathway, what's necessary from the cell side to really say what's going on. For this sort of study, from my line of work, there's the two sides of the coin. I think set of amino acids X is important. I took them out of protein A. Protein A stopped working. They are important. I can say pretty conclusively they are important. Are they the reason protein A makes a branch? I can take protein B, which is really, really close, like all of these ones that add sugar to the end instead of a branch. Why don't I just put those amino acids in those? See if they start making branches. Because if they do, those amino acids are responsible for branches. If they don't, those amino acids are important for the branching enzyme, but they are not the whole story of why that branching enzyme makes branches. And those are different answers, and those are different studies, and those are different places for that study to go. So the next step I would do here is take one of these other proteins that he shows in the cartoon that don't cock it to the side. Take one and just engineer the amino acids. You just outlined all of the ones that you can't get rid of, and all of the ones that you can get rid of but it hurts it, and all of the ones that you get rid of and it doesn't matter. Like, you just outlined all of them, which means you made a protein, at least this many mutants. I guarantee you probably tried at least a protein with a couple combinations here. Take all of them, take the whole bundle. Put them in a different protein that you already knew does the same exact chemistry. It just doesn't do it in the same order or the same direction or the same stereospecificity. Does it start making branches?
What do you mean by stereospecificity?
Right. The orientation of the thing, of the product or substrate. How selective are you and how strict are you in that it has to be a specific shape? I don't know the Latin basis for stereo, but like a stereo system where it's giving you music from different directions. Like two different directions.
Yes.
Stereospecificity is essentially like the shape of the thing, like how you're interacting with it from multiple directions, its shape, how does it fit somewhere? Some things are very, very stereospecific because they have a tiny, tiny little pocket that you have to weasel something into. Those things are stereospecific because
I know all about those things.
Only certain things fit there. Some have
more than you'd think.
Not a pocket, but an active site cleft, where it's just solvent exposed and things can just rest. those are often not stereospecific because it doesn't matter what the top shape is. As long as the bottom's resting fine, then it doesn't matter what everything else looks like. So stereospecificity is incredibly important here because that's why you have to grab the second glucose in such a way to angle it to just have the branch. So all of these enzymes are very stereospecific because they need a specific set of sugar chain to do their chemistry. And if you can change that and say, okay, well, protein A and B, you weren't special before. Can I make you special? And the answer is yes. Then you could apply, well, you don't have to stop protein A and B. Can you do it any sugar transferring molecule? Can you make it start branching? Can you make things that aren't sugars start branching? Can you, this goes on and on for the both academic purposes and industrial purposes. You unlocked a key. You now know that if you put this in a sugar transferring molecule, it can start making branches. Branches are a big deal. Branches are hard to break down. Branches attach to different receptors. Branches do this and that and the other thing. So they call it a signature motif. Allows for the formation of a branched sugar modified product, which may be useful for a signature motif to bioinformatically distinguish branched chain forming UGTs from those that directly glucosylate various substrates. If you got a signature motif, not only could you engineer it into things, but you could comb giant libraries of sugar moving molecules and say, oh, it has this set of 10. It probably makes branches. Nobody's ever characterized it before, but we can guess that it makes branches, so we need it in our lab because we need branch-making molecules. If you can magically find the right signature motifs, it changes fields if they can put it into something else and it works. Otherwise, they just figured out how this enzyme works, and that's neat. And you could use this specific enzyme in an industrial fashion and know a lot about how it works, which is cool and very helpful. But if they found it and it's got broader implications where they found a way to make other things make branch chains, they just changed quite a bit. But I think that is a very obvious biochemical question to make that they didn't address.
Oh.
So I don't know if that study's going on right now. I don't know if they already tried it but don't want to talk about it because it was just a bunch of negative results. I don't know if that's a separate paper from this lab when did this paper come out? This came out in 2019. There might be a paper that came out 3 months ago that is that exact study, and I just don't know about it yet. It's hard to know. But to me, as someone who has been trained in the kind of biochemistry that these people have been trained in, that you get crystal structures, you pour over the crystal structures a bunch, you make a bunch of mutations, you see what happens to the kinetics, you release a paper. That pipeline, I don't know why they didn't talk about it. I don't know why it wasn't mentioned in their conclusion section of future work would be such structure-guided efforts offer the potential for altered pathways of steviol production to generate tailored variants of this non-caloric sweetener. Yeah, that's great. But you're talking about altering the pathway that exists. Why aren't you talking about pathways that you could invent?
So because they've found this out, are we going to get more less bitter stevia?
Possibly. Because...
Give me my candy.
Because if this helps make Biosynthesis A easier to make, or better, or more viable, or we know what conditions it now needs that the plant likes it because we need to optimize conditions for these proteins, something like that. Yeah, it could make it easier. Right now, it seems like the mogrosides from monk fruit are a better alternative because they don't know the entire pathway for this. As long as there's a missing step. As long as there's a missing step, then you still have to go to the plant.
The missing link in candy evolution.
Yes, they are the missing link. So I don't know where this is going to go. For the time being, because they didn't show that they could engineer it into other things, I don't know if it's important enough to actually revolutionize anything. If it does, and you can put it in, you might have, so imagine, if you will, a vat, like a big vat of...
Like the one that made the Joker?
Yeah. Actually, yeah. Of 100 liters or 500 liters or a million billion liters.
So big.
Vat is just stirring around, and you have some sort of culture in there, be it bacterial or eukaryotic or something. and you have the reaction going on that this special enzyme here, the branch enzyme, does. And you have it swirling around at like 50 degrees Celsius.
That's hot.
If I could make it, yeah, it's hotter than body temp. It's not that hot. If I could make it 70 degrees Celsius.
That's even hotter.
Just by basic chemical kinetics, the chemistry will go faster because it is hotter, and hotter chemistry goes faster. But let's say special enzyme starts to lose activity at 60 degrees Celsius. So by the time I hit 70, sure, the reaction's going faster, but I have less enzyme hanging out actually able to do the job because it's starting to break apart. I haven't actually gained anything. But I could have a glucosyl transferase. I could have something that moves the sugars around, great, and be perfectly happy at 70 degrees Celsius. But it doesn't do the branched chemistry. I could take that enzyme and put all of these from figure 4, all of these residues in, engineer all of those amino acids into that new one, and it make branches now, even if it's just half as good, or maybe it's 20% worse than the native enzyme that does it because you haven't really optimized the whole thing. You just have optimized the active site. But then I can crank it up 20, 30 degrees Celsius. I've now gained a much, much better VAT reaction because I had a protein scaffold that I could make do the chemistry I wanted to. Potentially, that's what they did. But they didn't do that in their study. They didn't try that. Or they just know all of the ways to break this enzyme, which is neat.
I would break you.
I will break you. But it's not that helpful. Well, it's not that useful. It's helpful in that it's a neat discovery because we didn't know how it worked before, and now we do. But if we can't engineer it into other things to kind of proof of concept make new enzymes with it, then you knowing how this enzyme does it, it's just purely academic. And it's an academic paper from an academic lab, so that's fine. And that's why they get funding, and that's why they release papers, and that's why they're successful. But as far as the potential to revolutionize anything, this could just be a quaint little paper that falls into obscurity. Or it could be really, really, really helpful to the non-caloric sweetener industry, which, you know, as long as we keep baking and drinking coffee, it's a huge industry.
I'm a big fan of the industry.
Yes. So I guess we'll see. This was 2019. We'll see if anybody ever makes anything from this. It's possible, but I don't know.
So that's neat. It doesn't feel as groundbreaking as the last paper.
It doesn't feel as groundbreaking because it's not 6 different groups across the world and 30 authors. But
is it secretly as groundbreaking?
I mean, the other one sounds groundbreaking because it's an oral male contraceptive that's non-hormonal, but
this is just a non-sugar
does that really revolutionize the contraceptive industry if we already have a bunch of different kinds of contraceptive and we already have the societal block that the pill is for women? Like, it feels like it could be revolutionary, but even releasing that contraceptive might not matter at all. It might be a complete flop because society just doesn't care.
Yeah, but it's so cool.
It is so cool. And this is, we can make stevia the most delicious, sweetest, easiest to make non-caloric sweetener. That's, I don't know the actual market numbers there, but that's not no money. That's a lot of money. How many things have fake sugar in them?
Me, for sure.
Yeah. Think of the sodas, think of the baking, think of the candy, think of coffee sweeteners. Like,
I'm always thinking of these things.
Imagine if there was a one, like the breakout market one, that was like, I don't know, the Google, like the name that people, when they think fake sweet, like Band-Aid or something, like when they think fake sweetener, they just say this word because they don't even remember what it's called. They just know that it's that thing.
Here's what I want from the food science industry.
Yeah.
I want food so delicious and so low calorie that humans become just like pandas. And we're just always eating this low calorie thing all the time. That is my absolute dream. That is the perfect life.
There'd be no war. Everybody too busy eating.
I gotta eat, man.
Can't shoot and eat at the same time. That is beautiful. I don't know that that's possible.
Isn't that what this study is showing?
They don't.
Aren't we going on to a panda society?
They don't really mention pandas in this little blue box that says significance.
I feel like the panda's implied. It doesn't have to be specifically mentioned, because we won't actually be pandas. We'll be just humans going at it.
That is how I would mime going at it. Were I the type to mime such a thing?
It was the 2 eucalyptus leaves that I'm bringing up.
Of course.
It's not anything else. I'm just talking about eating. I want fake meat, low calorie fake meat that's still super delicious, and I want 0 calorie candy. And you'll give it to me with science?
Soon-Gu Lee will, from the Jezebel Lab.
I'd prefer all over you.
Poor Iden Solomon.
And nobody cares about Solomon.
He doesn't have a funny name or one of the important authorships.
So is that everything? Is that
Yes.
Today's group meeting?
Yeah, that's about the long and short of it.
Well, that's pretty neat. I
It
like
is
this
neat.
paper.
It is neat. And it's digestible. And it is...
But
I actually didn't mean to do that. It's the kind of paper that I have read a million times. And they have a particularly good setup for it. I think they walk through their story really well. Very straightforward, very well fleshed out. My only issue is that I wish they had mentioned the we want to put this in new enzymes, but whatever. I'm sure that's happening.
Thank you for bringing this forth this week and for explaining it to me and helping me and them get lay.
Get lay.